Okay, so we're going to continue talking about the antibodies and antigen. And before we move on, the antigenic determinant, it's more or less clear, right? So before we move on, we have to figure out how cells, I'm kind of debating, well, let's talk about antibody structure first, okay? So when we talk about antibody structure, you have to appreciate that not only antibodies that are produced by B cells, but plasma cells, but also various types of receptors on the B and T cells. Let's put it in the writing form. Antibodies, B and T cell receptors share basic structure. Okay? What I mean by that? It means that there are differences, but the concept that I'm going to explain is the same. Does that make sense? So all these molecules, antibodies, B and T cell receptors, have fairly similar parts. Sorry. So we're going to start with the colorful ones. Okay? Okay, it's not to scale, so don't be too critical of my drawings, please. Okay? Wait a minute. Just a little correction here. Here's the structure of antibody. So it's like a fork. You see the similarity? Do you see the similarity? It's a fork. Okay. So this fork consists of how many, and it's a protein. Okay, it's a protein. So how many chains can you see here? 4, right? You see 4 chains. This is light chain. This is heavy chain. This is heavy chain. And this is light chain. Okay? Light and heavy chains. Are we clear on that? Awesome. Next, you see the colors. Technically, I didn't have to put green and red, like different colors. The point is, black part, okay, this part is called FC fragment. These parts, the colored ones, are called FAB fragments. So you can see that so there are 2 FAB fragments and 1 FC fragment. Each FAB fragment consists of a part of the heavy and part of the light chain. Does it make sense? Huh? Each FAB fragment consists of a piece of light and piece of heavy chain. That's it. That's just, okay. We're on the same page. Now, why are they called FAB and FC? FAB, well, F stands for fragment. AB stands for antigen binding. FC stands for constant. Does that make sense? Okay? Now, when you use fork, how do you use it? So, okay, you stab with prongs, right? You use handle to hold it. Now, if you would look at different types of forks, and I looked several times, there's like salad fork and I don't know anymore because for me they're all the same, but they look different, okay? Some of them have like 3 or 4 prongs. Some of them have bigger, smaller. But the handles it's prongs that are different. Handles are pretty similar. Does it make sense? In case of antibodies, the handle, the Fc fragment, Fc fragments are pretty similar between all antibodies. There are some differences, but they're not conceptually important for us now. What is different is FAB, okay? Antigen binding. If you take antibody that recognizes one antigen, an antibody that recognizes another, their FAB fragments will be very different. Does that make sense? Now my question for you will be very simple. How many antigens are there? I think you underestimate. Well, I mean, how many pathogens are there, right? And how many proteins in each pathogen? In DNA. And they're all different. So we talk billions probably. Maybe more than that. Different types of potential antigens. So antibody is the protein. What is the process of protein synthesis? What does it start with? Like very first thing, protein DNA. So each protein is what? Should be a gene. So billions of different antigens means billions of different types of antibodies. Billions of different genes. Let's assume that one antibody is 200 amino acids. How many nucleotides you need to encode one amino acid? You need 3, right? 3. So 200 amino acids means that the gene has to be at least 600 nucleotides. If we assume billion, it means 600 billion nucleotides only for the genes that encode antibodies. The size of the human genome is 200 times smaller. So definitely we do not have a gene for each and every antigen. It should work some other way. And here we come with a concept of VDJ recombination. I don't know if you've heard about it. That is a complex thing, which actually is complex only when you read it in like 1,000-page immunology textbook. It can be complex, but I want you to understand the idea, the mechanism, the very big, because that's what you need. If you need more, you will read, okay? Here how it works. You have 3 types of genes. In fact, you have more than 3, but we will focus on these 3 types. You have V genes, you have D, and you have J. It's a group of genes, like a cluster, okay? Say you have 3 J's, you have 4 D's, and you have 5 V's. Are we clear? Is that understood? you have 5 V genes. All V genes are very similar. Very, very similar, but not identical, but similar. All D genes are similar, but not identical. All J's are similar, but not identical. Are we cool so far? When antibody is produced, it is the FAB, this fragment, is the combination of a V, a D, and a J. One gene of each. For instance, 311. And it provides the unique structure of that protein. If you will combine 512, it's going to be a different structure. So every time you combine these 3 genes, it creates a different structure of antigen binding fragment. Does that make sense? No? Okay. Let me find my keys. You can look at yours. So I have 3 keys that look very, very, very similar. They're different though. Okay? They open different locks. If you look at the keys, we will see that each key has pretty much the same elements. Some ridges, some, I don't know, crevices, whatever. Okay? You can see those like little things. They look similar. In size, they are similar. But what's different is the arrangement. That makes sense? It's a combination. You may have 5, 6 different elements, and combining them, you can create a huge number of different keys. Same here. We have something like 47 V genes, like 12D and 27J. I don't remember the exact number. But the point is, you can combine these different numbers. It's like a license plate on the car. Okay? Does that make sense? So, combinations will create almost infinite number of variants of this FAB fragment. Does it make sense? So each variant will have slightly different structure, which means it's going to bind different antigen. So we have cells, B cells, and we'll get to that. We have a capacity, our immune system has a capacity to recognize pretty much every possible antigen because of that infinite variation. Does that make sense? So that's what I was telling Brianna during the break. Imagine that we have a potential to make keys to every door on Earth. Does that make sense? We have all the elements that are used in those keys. Okay? Is that better? Clearer? You don't hesitate to tell me, no, it's not. Okay. Now, did you get the antibody structures? Now, I have to highlight that it's going to be antibodies, it's going to be receptors for B cells, it's going to be receptors for T cells. Now we need to figure out how B cells work. So just a quick reminder, B cells originate in the bone marrow, okay? They produced in the bone marrow and they stay there to mature. So how does that work? So you have a B cell that is naive, and you have to check it for one specific quality. Your immune cells, the B cells, What do they have to recognize? Huh? Antigen. All antigens, or there is a group of antigens that they shouldn't recognize? Which antigens they shouldn't recognize? Yeah, your own body antigens. Self-antigens. Does that make sense? Because if they do, what's going to happen? How do we call this? Autoimmune disease. Okay? So pretty much we ask B cells a question. Can they recognize own antigens and produce antibodies against them? It's often called self-reactivity. So there are two answers to this question, which are yes and no. If yes, what's going to happen to the cells? If they recognize your own body and start to make antibodies against your own tissues, what should we do with those cells? Huh? No, we don't want to keep them. What if they escape? Yeah. How do we call the programmed cell death? Apoptosis. So it triggers apoptosis. Does that make sense? If no, if they're not self-reactive, they go out and play. They go into the secondary lymphoid organs. And there in lymphoid organs, B cells become directly exposed to the antigen. They don't have antigen to be presented. They can just interact with an antigen. Does that make sense? Here what happens. So say you have 3 different types of B cells with the different B cell receptors. Does that make sense? That's a B cell receptor. That's the B cell. Each population of the B cell is called a clone. Clone 1, clone 2, and clone 3. Okay? Just the terminology. And they encounter the antigen. Which clone is going to recognize that antigen? Clone 2, right? So these guys, there will be no interaction here. Okay? Only clone 2 will recognize it. And then there are 2 things that happen to B cell clone that recognizes the antigen. Differentiation, which means when cells differentiate, what does that mean? They become more specific, yes, more specialized. And proliferation, which means multiplication, yes, they become more numerous. So far, you keeping up? Which one? Differentiation? Differentiation means that cell becomes more specific in its function. Proliferation, it becomes more numerous. So B cells, what they differentiate into? 2 things. 1, they form immunological memory. So they become a population of memory B cells. Cool? And second, they become plasma cells. And plasma cells start to produce what? There you go, antibodies. Does that make sense? That's B cell activation. So this step this step, when clone is selected, is called, you'd be awfully surprised, clonal selection. This entire step, when cells become differentiated and grow up, it's called clonal expansion. Does it make sense? The terminology-wise. That's just terms. You know, clonal selection, we select a specific clone. Clonal expansion expands. Okay. It's a great time to talk about types of antibodies. That's going to be not that exciting. I will tell you pretty short what you have to know about antibodies. There are 5 types. I'm going to start with the boring and go to the exciting. So the boring is, first boring antibody is IgD. Okay. Let me try and, I cannot make a straight line to save my life. Okay. IgD. Its function is to be a B cell receptor. Okay. So if this is B cell, this is IgD. Good? Now, important, you have to understand. So the antibodies that are going to be produced, that's one thing that I should mention. Antibodies that are going to be produced will be like this. Does that make sense? So they can recognize that same antigen. Does that make sense? Okay. So IgD is the B cell receptor. Next one is IgE. It causes degranulation of mast cells, sorry, and basophils. What it will be mostly responsible for then, if it causes degranulation of mast cells? Inflammation, but IgE, mast cells are in the connective tissue and they're responsible for some unwanted immune reaction. You're right, it's an inflammatory reaction. They release histamine, which is going to be when you take antihistamine drugs. Allergy. So it's pretty much, it's the antibody that mediates allergy. That's, you have to know that. Okay. IgA is a secretory found in mucous membrane. Let's call it mucus. Okay. Where are mucus? Where is mucus? Nose. Respiratory. Stoma, yeah, all digestive tract is mucus. Reproductive organs have mucus. Wherever there's mucus, there's IgA. It's like a first response. So it's adaptive response, but it's first response. Okay, it meets the pathogen at the mucous membrane. Does that make sense? They have quite an interesting structure. They're dimers. So 2 molecules, they stick together. There is another secretion in which IgAs are extremely important. You can go ahead. Seminal fluid contains IgAs, but trust me, when you will figure out what secretion is that, you realize not that important. The vital secretion, exogenous secretion. Oh no, it's not vital. I mean, it's vital, but not that vital. Huh? Other people depend on it. Really? Huh? No. That's not endocrine, exocrine secretion. Other people depend on it. Usually it's one person that depends on it. Breast milk. It's exogenous, it's former sweat glands. That's how we protect our young for like 6 months. For 6 months in their lives, they're protected by mother's antibodies. Okay? IgM. These are first responders in the blood. Okay? This immunoglobulin M is the first molecule that's produced in response to the infection. First antibody that is produced. First response. And they form pentamers. So they're gigantic. The complexes are huge. Okay. And finally, IgG, immunoglobulin G, which is about 70% of all antibodies that you produce. It's the most common antibody, can perform a variety of functions, and it's also, let's call it most common, and it also transplacental. Okay, it can cross placenta, which confers the additional immunity to the fetus. Okay? Yes?
Okay. Why does the GI tract of the infant break apart that protein and make it not work?
Why immunoglobulin is not
Inactivated by the stomach.
my strong suspicion is that since mother produces IgA and her intestine, and it's in the mucus, I never reached into it. It's a great question, by the way. I suspect that it has no available trypsin, chymotrypsin, or elastase attack sites. Does that make sense? So just the enzymes, the proteases of the digestive tract, they just don't have the site, the specific site, to cleave the protein at. Does that make sense? That would be my strongest suspicion. Okay. And that's probably, maybe the fact that it's dimer contributes to it. Also, you have to appreciate those I'm not going to ask this on exam, just to let you know. Heavy and light chains, they connected to each other by the disulfide bonds. So I believe even if you will introduce 1 or 2 cuts, the entire structure is not going to fall apart. Does it make sense? Like if you drill the hole in the wall is not going to crumble. Okay. Okay, how they work. So what they can do first, we're going to call it mechanisms. One mechanism is to opsonize. Okay. Remember, we already talked about opsonization. Pretty much you have a certain, like a pathogen, okay, and antibodies gonna surround it and make it more attractive for phagocytes. Does that make sense? So that will increase, opsonization will increase phagocytosis. They will make it like a ketchup. You put ketchup on the burger and it tastes better. Right? Another mechanism is agglutination. Let's say you have several cells, and when antibodies recognize it, they stick the Fc fragments together. Okay, can you see that? And they form a clump of cells. For instance, that's the mechanism by which the blood incompatibility works, the transfusion reactions. In case of microbes, agglutination, when there's a big clump, it is more quote-unquote noticeable for phagocytes. It also restricts the mobility of the microbes. Does it make sense? Mechanism that is very similar to agglutination is called precipitation, and it works not for the cells but for the molecules. So let's say if you have molecules of tetanus toxin, then antibodies against these molecules will stick the Fc fragments together, clump the molecules, make a larger sort of assembly that then, bless you, that then will be phagocytosed. So in essence, agglutination and precipitation are very similar. Agglutination works with cells. Precipitation works with molecules. Does that make sense so far? Okay? Now another mechanism, another mechanism is neutralization. This mechanism works practically exclusively for viruses. Well, let's put it this way. It works best for intracellular parasites. And here's the example why. So you have a virus, okay, and antibodies stick to it. Virus has to do something for infection. Where does it have to go? In the cell. And to get into the cell, what does it have to interact on the cellular surface? On the cellular surface. Receptor. And if the virus particle is all surrounded by antibodies, can it interact with the receptor? No. So nothing happens to the particle. Does that make sense? Nothing happens to it. Just cannot, it's stuck. Okay? And the fifth mechanism, I want to remind you, it's a complement activation via classical pathway. I'm not going to picture this, we've talked about it, but these are 5 mechanisms for antibodies to work. There's something that antibodies, with all that spectrum of activities, something that they don't do. Can you recognize what they don't do? Yes, they're not destroying anything. You see? They don't kill microbes. They don't kill cells. They tag. That's what they do. They tag. Okay? Does that make sense? Bingo, yeah. Like, you have to take care of it. Okay, yes? Penicillin. People who have allergy to penicillin, they develop IgEs to penicillin. IgEs stick to mast cells. When a person gets a penicillin, penicillin binds to IgEs. IgEs cause degranulation. Person has allergic reaction. That's how it works. Yes, absolutely. Absolutely. It's not common, and we'll discuss the mechanisms how it works, but that can happen. You can have antibodies to medications. Now, a few words about the primary and secondary response. Come on. This is fairly straightforward, I think. Just a few words. So if this is, let's put down the graph for the primary and secondary response. This is going to be, y-axis is going to be the level of antibodies, and x-axis is going to be time. This is going to be the primary exposure. This is going to be the secondary exposure. primary exposure. Take some time to produce IgM first. It takes even more time to produce more IgG. Does that make sense? Remember, it's not to scale. You can see that antibody levels decline. How much they will decline, it all depends. Okay, it's the hypothetical situation. Then you get exposed to the same antigen, same pathogen second time. What's going to happen? IgAs, I'm not focusing, IgAs are going to be produced and they're going to go to the mucous membranes. You're absolutely right. But the main component of the response is IgG, we have to admit it. So what's going to happen to the IgG levels after the second exposure compared to the first? Quicker and more. It's going to be more like this. Does that make sense? IgMs are still going to be produced, don't get me wrong. Okay? So you can definitely see the difference in exposure. Now, how can you get exposed to the antigen? Okay, vaccines. They're designed by Mother Nature or humans? Humans, so they're artificial, right? So let's figure out the types of humoral immunity. Vaccines. Artificial. What would be the opposite of vaccines? What would be a natural way to get exposed to the antigen? Yeah, infection, right? Actual infection. So this is natural. Now in this case, your immune system, does it actually deal with an antigen? Does it recognize the antigen? Yeah, vaccines contain antigen, bugs contain antigens, so it's active, right? Your immune system is exposed to the antigen. Vaccines is gonna induce active artificial immunity. Infection is going to induce active natural immunity. Is that clear? Can you get protected without being exposed to the antigen? How? Breast milk. Is that natural? Of course. Is there any way to get protected without exposure? Not through the breast milk? Genetics is, inoculation of what?
Similar pathogen.
It's going to be active. You're going to, without exposure. It's going to be exposure. Can you just take immunoglobulins? Yeah. Immunoglobulins. You take immunoglobulins and you inject them. Does that make sense? Just, you can isolate them from the person. You can artificially get them from the horse. The anti-venom, if you get bitten by a snake and they inject anti-venom, that's produced in the horse. Tetanus shot, not the vaccine, but the one that you get after the acute exposure. I don't know where it is produced, but that's antibodies. When you get bitten by, say, a rabid skunk, the site of bite, they make shots around the site of bite with anti-rabies immunoglobulin. It's artificial. Does that make sense? Immunoglobulin. They've got the antibodies. They had monoclonals, I think from the boy. that actually survived from Ebola. So pretty much isolated them. And that's going to be passive. I can absolutely guarantee that I will describe you a case and you have to recognize what type of immunity is that. Passive artificial, natural artificial, and blah, blah, blah. Okay? Now, in the last few minutes, we're not going to play that game about vaccines today, but next, on Thursday. It's pretty fun. It's going to be pretty fun. You will take it home. Okay? What I want to mention is vaccines. We're going to talk about vaccines, and that's only what I want you to know about vaccines so far. How can you make a vaccine? What are the options? Okay, part of the pathogen, dead pathogen, inactive. So, vaccine can be inactivated. That's Chris's proposal. Vaccine can be a part of the pathogen. It's called subunit. Do you know any inactivated vaccines? Huh? Which one? Huh? Which one? There are two. One in the nose, one shot. Shot, okay? So flu, polio, inactivated polio vaccine, IPV, also shot. Those are inactivated. We can't say dead because viruses are not live in the first place. Flu and polio viruses. It's a terminology thing. Subunit. Do you know any subunit vaccines? They're fancy, extremely safe, and horribly expensive. I don't believe Rota is the subunit. Huh? Hep B. It's awfully safe. Gardasil, I think Gardasil is subunit as well, yeah. Yeah, I think HPV is subunit too. Hep B is the first one. I mean, it's pretty much, it's given second day. I think second or third day after the birth. Okay. Any other way? Yes. Uh-huh. It's pretty normal. I mean, vaccine is not 100% effective, period. That's number one. Second, the so-called correlate of immunity, it's very fancy word, pretty much the parameter that is measured and tells you whether vaccine worked is antibody levels. In some cases, you may have immune response without having increased antibody levels. But it's some cases. So if you don't have antibodies, it's safer to give you another try. Does that make sense? Any other way? Okay, plasmid. You can take plasmid, stick the gene or genes into the plasmid, shoot it into the muscle, and those genes will start getting expressed. Okay, and proteins are produced and your immune system will make. There are no approved plasmid vaccines yet. WHO doesn't like the idea. Another way is to stick the genes into the some harmless bug like a virus. Okay? So you can inject virus will replicate, it's not going to hurt you, but it will also produce proteins of the pathogen that you are protecting with. For instance, you can take yellow fever, the vaccine strain of yellow fever virus, gut it out, throw in proteins of like dengue virus, and that recombinant construction will replicate, will reproduce, and you will get immunity against dengue and yellow fever together. Does that make sense? That's a whole different story. No, I mean, that would be great, but nobody would agree to that. Because Guess what? For vaccination, you gotta have a consent. I don't think mosquitoes will ask. You forgot about the fifth way. Anyone got ever oral polio vaccine? The drop of fluid on the cube of sugar. It's a live vaccine. It's also called attenuated, which means weak pathogen. Does that make sense? Pathogen that doesn't cause the disease. Examples are numerous. Oral polio vaccine, which is not inactivated. Flu mist. MMR. Yellow fever. 17D strain. Okay. There are advantages and disadvantages of each, and we're going to chat about vaccines and from the health. We're